Right, good morning, everybody. It's a really great pleasure to be here and to get the opportunity to present Xintela and our development programs using MSCs. Forward-looking statements, and you find more information on our website. So Xintela, we're a biopharma company focusing on stem cell therapy and also cancer therapy. That's our subsidiary to XINT. I will not talk about that today. We are a team of about 30 people based Medicon Village, south of Sweden. And we are listed on NASDAQ First North since 2016. Our research and development is based on a specific integrin marker, a cell surface marker, integrin alpha 10 beta 1 as a collagen binding receptor. And this integrin is really key in our development, and I will tell you about that. So we using it in our stem cell-based therapies and also for antibody-based cancer therapies. Highlights from our stem cell business. Now I will focus on the stem cell part. So we have developed a superior stem cell product named XSTEM, which is patent protected for all therapeutic use. We have over the years built up really strong preclinical data supporting XSTEM therapy. We have 2 projects in clinical development in osteoarthritis and in difficult to heal venous leg ulcers. And we have also built our own GMP facilities, so we produce our own product. So the collagen binding integrin was actually first discovered in my research lab at Lund University on chondrocytes. And then we made this interesting discovery that it is also present on MSCs, which is key here. And also some aggressive cancer cells, as I mentioned, have hijacked this stem cell marker and use it, and that's why it's also an interesting target. All right, so with this integrin marker, integrin alpha 10, we can select homogeneous, high-quality MSC preparations. So we can really solve this problem that was also mentioned before, that stem cell preparation, doesn't matter what source you use, but they are heterogeneous and they are more or less heterogeneous. So you have MSCs in there, but you also have other cells that we want to get rid of. And that problem we solve by using our integrin markers with antibodies and a selection technique. We can and then get homogeneous MSC preparations. I also like to mention that these MSCs also have these markers that are recommended by the International Society for Cell Therapy. But these markers that are currently used today are also present on the other contaminating cells in MSC preparations. And the lower picture there really shows you what differences makes for chondrogenic differentiation. You see the blue cells there. When we make a pellet out of these MSCs, differentiate them to chondrocytes, we can see they really express high amount of the cartilage molecule type 2 collagen, while the other cells labeled there, when we make a pellet out of that, they do not have that capacity. So it's a clear difference here between cells that you have in that preparation. Oh, sorry. Oh, what did I do now? Okay. So XSTEM then, our stem cells, we isolate, so these are allogeneic MSCs, we isolate them from adipose tissue. It's a great source, plenty of MSCs available, so we can actually get massive amount of cells with limited expansion. And the XSTEM, we have really shown that it has great both functional and regulatory advantages. And one important point is really this reproducibility that we have with this technology, that we get constant high quality between batches, between donors, and so on. And also, which I will show a little bit more later on here, we have improved differentiation capacity, improved immune modulation capacity, and also, which is important, improved homing to the damaged tissue. This has been published. So we have XSTEM in clinical development in osteoarthritis and also in difficult to heal leg ulcers. We have also completed a preclinical program in ARDS and also are looking into additional indications. And we also have a program in veterinary medicine. So I mentioned we have built our own GMP facility, a great advantage, of course. So it's have a license now since 1.5 years to manufacture ATMPs for clinical studies. We manufacture our products developed at Xintela and also together with partners, and as to control your manufacturing, of course, you know, it reduces production costs, risks of delays, and all that. So that's a great asset. And we also now can start to offer contract manufacturing of ATMPs for clients. We heard about osteoarthritis earlier here today. It's a huge medical problem and also, of course, a huge market, and that we have no treatments today. Many of us end up with a joint replacement. We have investigated the selected MSCs in horse models, actually in two models. So we've been able to show both safety and efficacy with integrin-selected equine MSCs. The first study we did was together with Lisa Fortier at Cornell University, where post-traumatic OA is induced in the ankle joint, and then we inject MSCs in one joint, and the other one is the control. And you can see there in the middle panel there, the normal cartilage there on the top, red, and then after 6 months in this model, degraded cartilage without treatment, and that significant less cartilage damage after treatment, and also less bone sclerosis. This has, together with a lot of other results, has been published in American Journal of Sports Medicine. We also did another study to be able to look at function, to look at lameness. So we did another horse study together with a group at Copenhagen University. So here it is, post-traumatic OA in the carpal joint. And so 18 days after OA was induced, MSCs were injected in one group of horses, and the other group was left untreated. And we could see the black line there, we could see significant less lameness in the treated horses compared to the untreated. Also significant reduction in pathological changes and significant increase in PGE2 and IL-10. So then what's the mechanism of action of these integrin alpha-10 selected MSCs? Well, the cells that we used in the horse studies were selected then for a high expression of integrin alpha-10. You see the upper left there with a population that we selected by flow cytometry. So those were used. And then we compared this population of cells with MSCs with a low alpha-10 and also with unselected MSCs. So we can conclude that we have improved adhesion of the cells to the cartilage. To the lower left, you see adhesion to cartilage defect and subchondral damages also. And as I mentioned also before, improved differentiation into chondrocytes. You see the middle pellet there with high type 2 collagen expression. The upper one is the unsorted, and the lower one, MSCs with low or no integrin alpha-10. And to the right, we have also seen improved secretion of PG2 in the integrin alpha-10 selected MSCs compared to unselected. And of course, PGE2 is an interesting factor known also, among other things, to switch macrophages from an M1 pro-inflammatory stage to an anti-inflammatory stage. But then we also wanted to look at homing and regeneration or differentiation in an in vivo model. So we label this is now human XTAM, the human cells. So we labeled them with iron and also with a fluorescence marker. So we could actually trace them by MRI and also look at the tissue, closer to the tissue. So we did this in a rabbit study where cartilage damage was made in the knee cartilage. You see that to the far left, you see that hole there where the damage is. And then we injected the labeled MSCs, and 4 days after injection, we could actually trace them. We could see, if you see that second picture now, where you see these black dots. So that's the MSCs that are labeled, that are actually home to the damage. Then when we section that tissue in that area, the right picture, you can see this is now full of labeled cells. So we have the MSCs there. We can also see that they have differentiated to cartilage-like cells because they co-localize with aggrecan, the cartilage matrix, and so produce and directly participate in the regeneration of this tissue. So this is really unique data and really good news, and this was published in May this year. So expected mechanism then of action for XSTEM in our OA patients is homing to the damaged tissue, differentiation and integration, and directly participating then in the repair, and also immune modulation and anti-inflammation, which have a positive effect on the whole joint, of course. And below there to the left, you see adhesion to OA cartilage. We see trillionish differentiation in the middle, and to the right, where XSTEM actually switch macrophages to an M2 phenotype. So our preclinical results really support our aim to develop a D-mode, a disease-modifying OA drug, to stop further development of OA, regenerate and heal the cartilage, and also reduce the need for joint replacements. So we have a clinical study ongoing in Australia. It's a first-in-human phase 1/2A study in patients with knee OA, grade 2 and 3. This is a dose escalation study, 3 dose levels, and with an optional dose expansion up to 54 patients. So it's a single intra-articular injection. So we have safety and preliminary efficacy assessment every 6 months and up to 18 months. And so we expect early safety data in the end of 22 and early efficacy data in 23. And to date, we have the first cohort dosed, and we'll soon start the next. So a few words also about our second program in difficult-to-heal venous leg ulcers. So you see this is really, to understand, a really painful disease and affect a lot of people, and we have no treatment available for these patients today. We have also here done a really excellent preclinical study in the pigs where we looked at XSTEM healing of wounds. And to the left there you see the skin, normal skin from a pig, and to the right is the regenerated skin from XSTEM. And from expert looking at this say that this really great structure that we see. This is 14 days after the treatment. So we have right now, or just now in October, started our clinical study for venous leg ulcers patients, and this will be 12 patients, single dose, administered topically on top then of the ulcer. So safety and preliminary efficacy assessed after 10 weeks and also after 6 months. And we expect to have early safety and efficacy data in the end of 22. Just mention a little bit about our ARDS study, the preclinical study. I mentioned that we have now completed with really great data after intravenous injection with MSC. So we have really significant improvement of clinical parameters and anti-inflammatory and immune modulatory effect, and significant less blood coagulation seen, and as you see there in picture, also less lung injury found in the lungs in the treated animals compared to the untreated. So now we're really looking forward to some near-term development milestone here, as I mentioned, in the end of the year to have early safety and efficacy results from our venous leg ulcer study. And next year, final safety and efficacy result from this study. Safety results from all dose levels from the OA study, and also early efficacy signals. And then also we now ready to expand the clinical development of XSTEM also into other indications. And we look for partners, also like to mention that, to advance further development of XSTEM. And what we can offer is really a very well characterized superior stem cell product platform, XSTEM. And as I've shown, and we also have more really strong preclinical data. So we know our cell, we know how, and we really have great identity of our product and a consistency, as I mentioned. And we're now, as I mentioned, in clinical development, and we have our own GMP facility to manufacture the product. So with that, thank you very much.